All right, let's try this again. I'm not sure why the audio didn't get sent to you. I recorded it multiple times. So we're going to try this approach. This is me recording on a Zoom meeting with just myself. And hopefully that will get the presentation to you because I recorded it 2 or 3 times. I just don't know what it is or what happened to it because it was there. So sorry for the trouble. Let me go through this presentation again. So basically my topic I picked was looking at paleopathology and proteomics. Basically, how are those two related in terms of ancient Egyptian studies? Come on, think. No, we're going to do this. Okay, so overview. So what am I going to talk about in this presentation? We're going to talk about, first and foremost, what is proteomics? Why is it important? Why should we even use it? What are the methods for it? And how does it relate to ancient Egyptian studies? As well as the weaknesses of the methodology, because no technique in the lab is perfect. They all could use some improvement. And then, of course, we have our conclusions and our sources. So first and foremost, an introduction. So there's a few definitions on your screen, just kind of show you from an umbrella approach of where we're going. So we got from the broadest to the most narrow there. So we have paleopathology, which is the broad field that has a whole bunch of different methods to study disease and pathological conditions in ancient human remains. This is obviously not just specific to ancient Egypt. This could be any ancient culture. Then we have the proteome, that's the proteins being that are produced by the genome of the person. So that genome encodes what protein should be produced. And then we have proteomics, which is the study of that proteome to determine the composition of the amino acids, because of course proteins are made out of amino acids. So by knowing which ones are there, we can figure out what proteins are there. And then paleoproteomics is a term I've seen in several papers I read. It's basically the application of proteomes, the study of the proteomes and how they relate to ancient peoples. And the reason this is important is because ancient Egyptians faced struggles against diseases just like we do today. And there were people who did practice medicine back then, but they didn't have the resources and medical technology we did today. So diagnosis was kind of hit and miss. And of course, they didn't keep records exactly like we do today, where every time you go into a doctor, they take down detailed notes of what happened and what you said and things like that. So when we have these medical papyri, we don't know exactly everything because there are missing pieces of the papyrus. And of course, they didn't write everything down like that. So if we can use kind of a modern technological scientific approach, we can kind of fill in some of those gaps and determine what was happening to the ancient Egyptian populace and their royalty, depending on which mummy you're looking at. What kind of diseases were they facing? Was it an endemic situation or was it just one-off? Did only one person have this situation? And the reason we want to study the protein specifically is because proteins are important to countless biological processes. So if you can determine which proteins are present based on the amino acids, you can figure out roughly how the disease worked or what was there, because a lot of proteins cause diseases. Like if they're misfolded, you can get really bad diseases. All right, so here's some methods. So say you want to do a proteomic study on your mummy. You have a mummy. Cool. Okay, so first things first, you're going to get a source of your protein. Typically, this is, like if you're looking at human remains, this is going to involve some sort of biopsy. That's one of the first negatives of this methodology is you do have to kind of destroy parts of what you're looking at to get the protein extracted. Because you want to collect through a biopsy, typically done with a syringe. I've seen a lot of papers use amounts of like 50 milligrams of like bone or muscle, things like that. And you're going to extract the proteins through lysis buffer. You're going to homogenize it, centrifuge it. And when you centrifuge it, you get a supernatant and a protein pellet. Depending on what you're looking for, you may then spin that supernatant down again, get another protein pellet. It kind of just depends on what you're doing exactly. Then you're going to reconstitute that protein pellet in SDS buffer and run it on a gel. The caveat to this being is ancient proteins don't always show clear separated bands on a gel. So if you're trying to use a gel to prove, this weight, this protein weight is there, so we think we have it, it's kind of hit and miss because proteins do degrade over time, just like DNA. So then proteins are very large molecules. To make them best suited for running through a mass spectrometer to determine what is there, It's best to cleave them and turn them into peptides, so little segments of protein, basically. And they're going to run those through mass spectrometer. That's the star of the show in terms of proteomics. That's where you're getting your actual information from, because mass spec is really great for determining what you have and how much of it you have, things like that. It can determine elemental composition, it can determine amino acids. So if you have an unknown, basically, which is what essentially you have, and you're running it through there, the larger, heavier things are going to fall off first, so it goes through these columns, and then the smaller ones are going to go further, just like in an SDS gel. So here is a picture of a mass spec. Here's the link where I got it. It's a Thermo Fisher one. I've used a lot of Thermo Fisher stuff in my day, so that's why I picked that one. All right, so here's some really cool studies. This is not all inclusive for this short presentation. Obviously, I couldn't include every single thing that's ever been done, but I picked these four because they stood out to me, because of what they found out or what they're trying to do and how it could be improved. So first things first, in 2016, this study group, they used skin and muscle tissue from 3 separate ancient Egyptian mummies. They were about 4,200 years old. And so they did the biopsy method. They did collect these muscle tissue through syringes and things like that. And they used different points of the body for comparison. So they processed the proteins in a standard fashion, mass spec the whole 9 yards. They did high performance liquid chromatography mass spec, and they found a total of 230 proteins, including collagens and keratins. That makes sense. We're looking at skin tissue. Skin has collagen. But what was most interesting to me is they found proteins that show there was use of the innate immune system in 2 of the mummies. And one of those also had the presence of proteins that are associated with severe tissue inflammation that happens after an infection. So they are speculating that this individual probably died of an infection, which is obviously something that was very common in previous antibiotic times, although it can still, of course, happen today. So that was really interesting to me, because you can look at it from a backwards point of view and you can determine a cause of death thousands of years later. Like, that's awesome. The next study in 2020, so we had a non-invasive method to extract proteins from a young female that was from the Old Kingdom. So this was interesting because one of the concerns in this is, in your data collection for this method, is, are you actually getting proteins from the mummy? So this non-invasive method showed a way to separate endogenous, so the mummy material, from anything that was applied to the mummy's skin, like during the mummification process, like any sort of like resin from trees or something like that, because that can be a separate tagging protein. So that is a really good point to make. And this one also, they found some human collagen keratin because of what they did. And they also did possibly find probable fungal and bacterial samples. That one, it's kind of harder to guesstimate the age on, because again, we don't know contamination. In 2022, there were a couple interesting studies, the first of which used both non-invasive techniques like CT scans and minimally invasive techniques like the biopsies to study Takabuti, the Theban woman from the 25th dynasty. So this study is really interesting because, so they looked at her CT scan and they noticed some issues like in her midsection, she might have had a blow to it, and then put that with the proteomics method, and they saw from her muscle tissue in her thigh that muscle tissue had been activated for a long period of time right before death. You put all those factors together and they have this really probable cause that this woman was murdered thousands of years ago. And that is wild. Again, like showing the cause of death and something like that that's not caused by a disease thousands of years later is just insane to me. So I thought that was really interesting. Sad for her, feel bad, but it's still a really cool scientific thing, and show what we can do with proteomics. And then again in 2022, this group, another group, they used tape strips. So basically putting tape on the skin. They used a modern living volunteer, and then they used some mummy bones. They didn't specify the mummy in detail, just it was from the 26th dynasty, I believe, or 26th, I don't remember which one. I have the paper, but either way. So they collected this non-invasive way. So basically you take the tape strip and then you get the protein off of it. Yeah. It did show some proteins, but not all the proteins they suspected were there. And there is a cause for contamination as well. So it's a step in the right direction, but it's not perfect. But it's something we could build on for the future. All right, weaknesses. Again, not everything is perfect. First and foremost, protein extraction. Like I said, it's going to require destruction, biopsy. Something is going to have to get messed up a little bit. If you can get multiple extractions from the same set of remains to do compare and contrast, you are very lucky. This does not happen that often. Museums and governments are very, very protective of their mummies for obvious reasons. So getting access to that kind of sample is more difficult these days. Shotgun proteomic analysis, it does work kind of like PCR for DNA, where you can start with very, very small amounts of protein, and it gives you a pretty good result at the end. It can extrapolate from that, but the problem is it can't separate from contamination. So if you have contamination in there with your small amount of starting protein, you're going to amplify the contamination, and then your data isn't good. So that needs some improvements. Again, contamination is a big star here. It can be a huge issue, especially when we're talking about mummies that were discovered in 1800s. They weren't wearing gloves when they went through these mummies. We don't know if, like, if you take a tape strip on their arm, are you getting the mummy proteins, or are you getting somebody from the 1800s proteins because they were touching it without gloves? We don't know. So former contamination, current contamination, all that is a big issue. And then, of course, non-invasive samples are going to be more just for external sample collection. You aren't going to be able to, like, take a sample from, like, an internal of the gastrointestinal tract. Looking at the proteins in that could show you, did they have any underlying digestion issues? What did they eat? How was their nutrient absorption? Those are all things you could determine if you could look at those samples, but you're not going to get that from a tape strip on the femur, you know? So there is a lot of information we could still get, but we just have to work on methods that will appease everyone and not damage them. It remains. So conclusions. So this method of analysis is really important, like I said, to continue working on it and improving, because it can show us so much more about the mummies. It can show us if we can get a large group of mummies, and in a perfect world, we got permission to, like, look at their intestinal tract or something using this method. We could look and see, like, what was the nutrition across an area at a time period, like in the different social rankings. Because we all know, like, there was worms and things like that. Like, how overall were they faring exactly if we could do that? Ancient Egyptian medicine, we always want to know more about how they were doing, what they did, why they did it. Again, the papyri are not perfect. So by kind of working backwards, by looking at proteins and ancient DNA and things like that, we can actually fill in those pieces, learn more about how ancient Egyptians looked at medicine. How did they treat their people? Why did they treat their people this way? And it can also fill in gaps to the culture. And then, of course, this modern medicine, proteomics is important for modern medicine as well. If you get sick and you want to know why, proteomics is a great way to figure out what proteins you have that might be caused from an infection or who knows what, and that can help you get an answer. So by developing methods that are more precise to study ancient samples, which can be a little finicky, we can actually make modern medicine better for the people today. And then, of course, we need to continue to work on the non-invasive methods for sample collection, because I want to be able to get a full picture of the proteome that's both internal and external. I think comparison of those two would be really interesting. And here's a picture with the link here of Takabuti in her museum where she is now, that woman who was murdered and that I mentioned earlier in the show. So here are my sources. Yeah. Okay. So thank you for watching. I'm sorry about the weird recording problems. Hopefully this works. So thank you very much for your time, and I will talk to you soon.